The O-GlcNAc Centre is proud to present the SFU Canada Research Chairs Seminar Series. This bi-weekly series hosts 5 presentations this semester. For the Spring 2010 semester, the presenters belong to the Faculties of Science, the Faculty of Communication, Art and Technology, and the Faculty of Health Sciences. Today's speaker is Dr. David Vocadlo, Canada Research Chair in Chemical Glycobiology from the Department of Chemistry.
It's my pleasure today to introduce Professor David Vocadlo from the Department of Chemistry, who's going to give the next talk in the seminar series of Canada Research Chairs. David hails originally from Ontario, but he did head west where he went to across town, the University of British Columbia, where he obtained his bachelor's degree, his undergrad degree, and then he stayed on there to complete his PhD in bioorganic chemistry with Professor Steve Withers, and he obtained that in 2002. And it ultimately led to an award where he received the NSERC Doctoral Thesis Prize for his work. He then went on to UC Berkeley, and he did 1 year there with Professor C.R. Bertuzzi. And then David came and joined the Department of Chemistry here at SFU in 2004 as assistant professor and, of course, as Canada Research Chair in Chemical Glycobiology. So in just over 5 years here at SFU, David has made some major accomplishments, and he has received quite a bit of recognition for his research. So in addition to being a Canada Research Chair, he also held a Protein Engineering Network of Excellence of Canada Chair from 2004 to 2006. He is a Michael Smith Health Research Foundation Scholar, and he currently holds an NSERC Accelerator Grant. And these are awarded for the purpose of accelerating progress and maximizing impact of outstanding research programs. And so I think that speaks for itself on some of David's accomplishments. So David is now an associate professor in the Department of Chemistry here at SFU. He's also an associate member of the Department of Molecular Biology and Biochemistry at SFU, and an associate member of the Brain Research Center at UBC. So he's accomplished a lot in a few years. We're sure he's going to continue to reach great heights in his research. And I understand that he also likes to reach great heights outside of the laboratory and doing some rock climbing. So I think right now he's going to keep both feet on the ground. And I'll hand it over to David and his talk today, Recent Advances in Glycobiology and the Development of Chemical Tools. David.
All right, thanks very much, Lee, for the kind and embarrassing introduction. So I'd also like to thank Vessel and the organizers of the Canada Research Chairs Seminar Series for the invitation today and the opportunity to share with you some of the results from the laboratory of chemical glycobiology. And I think I'd like to just correct one thing in Lee's introduction, that while I would love to take credit for all of this, the accomplishments, I certainly can't do that. I work with a tremendous team of researchers, graduate students, postdoctoral fellows, and undergraduates. And they're the people that really do all of the work I'm going to talk about today. So I'd like to thank them, first of all. They're sitting at the back here. So, of course, I'd also like to thank the Canada Research Chairs Program for ongoing support of our research program here at Simon Fraser University. It's a great advantage to be supported in this way, and the recognition that comes with the award is beneficial in promoting the research in the wider research community. So, when asked to give a talk, a general talk to an audience with a diverse background, I think there are 2 motivating reasons that I really like to do this. One of them is, as I said before and alluded to before, is to promote the research of the laboratory and to highlight some of the excellent work that these talented people who are working in the laboratory to people and promote their research and their careers. And the second reason is really to promote a better understanding of glycoscience and glycobiology in the general community, and to emphasize the importance of research in this area of science. So I'm going to start off then to accomplish these 2 aims by giving a general introduction to glycobiology and highlighting some of the recent advances that are relevant to improving the standard of human life in the world. And then secondly, I'm going to go into details of our research program and talk about one particular project that's been the longest running project in the lab. I'd like to talk about all of them in the interest of exposing the work done by the people in the lab, but I'm not going to be able to do that. So I'll focus on one project. Okay, so, So to start off, I think it's beneficial to get an idea of the scale at which experimenters carry out their research. And when studying organismal biology and biochemistry, one can look, of course, at tissues and organisms. So for example, up here we have a brain, and I think we can all relate to brain tissue, the size of the brain. We all know about how large a brain is. It's up here in our cranium, and that's very tangible. But then when we go and we start thinking about cells, which are on the order of micrometers, these are actually really quite small beasts, and to observe them is quite challenging. You need to use either direct methods of magnification or indirect methods of studying these little creatures. And within the human brain, there are about 100 billion neurons. Okay? Now, that's, of course, an awful lot, but it's not just that many cells. There are, of course, glial cells which support and nourish these neurons. So the number of cells within the brain is much larger than 100 billion. So when we go then into these cells and we examine, we could look in detail at the inside of the cell, what we actually find is that the cells themselves are highly organized and contain many different compartments. So one can see that a cell, you can make an allusion to a cell being a city with all kinds of different neighborhoods in which you have, for example, the nucleus shown here, or the Golgi apparatus shown over here, which I'm going to mention a little later on. Now, within each of these neighborhoods, there are all kinds of different citizens of the cell. And these citizens are the major biomolecules that make up and constitute cells and confer life, the ability to support life, to cells. So shown over here, this is a detail of a protein obtained using X-ray diffraction methods. So this is completely indirectly observed, and you build up a model of the structure. It gives you an idea of the general fold in this cartoon model. And then when we zoom in even further into the detail of a particular region of this protein, what we see is the atomic positions of particular atoms indicated by the vertices and termini of all of these chains. And so what we're doing is we're going down from an organism to cells to organelles to the biomolecules themselves, and ultimately right down to the atomic coordinates. And so researchers work in all of these different regimens of size, and in our laboratory, we focus primarily at the atomic level using chemistry, and at this, what would be called the biochemical level, studying these biomolecules. We also do some cell biology to study the effects of perturbing these systems at the molecular level, and also go into looking at the effects on organisms as well, to a lesser extent. Okay, so to give you an idea of the scale of these things, I think it's nice to be able to get a tangible understanding of this. Consider for a moment, if you were a cell, then a protein inside of you would be a grain of sand. And the body that you were in would be 80 kilometers tall. Okay? So this gives you a sense about the diversity of sizes here that are investigating using various tools and methods. Okay, so the major biomolecules of the cell are illustrated here. There are 4 major classes of biomolecules. Nucleic acids, shown over here, illustrated by this iconic double helix structure of DNA. And this is, of course, it encodes the genome, and RNA is encoded by the genome, as are other types of nucleic acids, which are now becoming of greater interest and of importance. Then there are, of course, the proteins, and these act as the catalysts that enable processes of life to occur in the cell. In the majority of cases, these are passive encoders of information. The proteins themselves are the executors. They may read the code or be coded by this code and then ultimately go on and carry out various functions within the cell and organize the structure of the cell. and these are directly encoded by the genome. Then you have lipids, shown over here, and these are somewhat different in that they're not molecules that are attached directly one to another by covalent bonds, so they're not physically attached in a very strong manner, but they self-assemble into complex structures. And in fact, the plasma membrane that surrounds cells is composed of a lipid bilayer. So the self-assembly of these molecules results in the formation of this lipid bilayer. And the topic that I'll be discussing primarily are these glycans. And these are not directly encoded by the genome as the lipids. They are also not encoded by the genome. And these are assembled likewise from various building blocks to form very complex structures. Okay, so let's just take a look at some of these major classes of biomolecules. So we'll look for a moment at nucleic acids. Nucleic acids, there are 4 different nucleotides in both DNA and in RNA. And these assemble into linear structures, shown here, illustrated in this way. Now, these linear structures, in the case of RNA, fold up into quite well-structured species. Some of these actually have catalytic activities, and they're being found more and more to have all kinds of different functions within the cell. And DNA, of course, forms this classical double helical structure. Now, what's interesting, because you have 4 building blocks and nucleotides assemble in a linear chain, what you have is if you have 4 nucleotides in a row, you have 256 different possible sequences that you can generate. Now, if you were to have 5 of these nucleotide building blocks in a row to make a chain, you would have 1,024 possibilities. So while this is a significant number of different possibilities, it's really not a staggering number. And in fact, the limited number of structures that you can obtain from the assembly of these nucleotides have enabled the development of all kinds of different technologies that have been very important. So if I say the term genomics to this audience, I'm sure those of you from background of history or English literature even would have an appreciation of the term. It's penetrated into the popular consciousness, the term genomics. And that's really a reflection of how pervasive these technologies have become on human life. So for example, one can sequence DNA very easily. There are many, many sequencing machines, and they're highly efficient. And in fact, that's how many of the genomes were sequenced. Well, all of the genomes now are sequenced using these very sophisticated sequencing machines. Likewise, nucleotide sequences can be synthesized using chemical synthesizers. Chemists have worked out elaborate ways to make these linear structures in a very simple manner. So I can call someone up and say, I'd like the sequence, I send it over to them, and they synthesize it, and 2 days later, I get it in a package and I can use it, okay? Likewise, if I have some DNA that I don't know the sequence of, I can send it off, and paying about between $10 and $30, I get the sequence back in a couple of days, okay? So these technologies are pervasive and very simple to use now. The enzymes that can be used to manipulate DNA sequences are readily available. So it enables a rapid manipulation of DNA and enables many, many different technologies and advances in biomedicine. Likewise, there are multiple good chemical tools to manipulate nucleic acids within cells and organisms, although there is, of course, scope for improvement in this region. Okay? So all of these advances have really stimulated fantastic advances in biomedicine, and the development of therapeutics now rely very heavily on these technologies of genomics and the chemistries related to that. Okay, now if we look at proteins, proteins are composed of 20 different building blocks. They likewise assemble into linear structures. These linear structures assemble into secondary structures, which in turn assemble into proteins. Okay? These proteins are very well-structured species, and you can actually determine the structures of these using sophisticated biophysical techniques such as X-ray crystallography and nuclear magnetic resonance. Now, because there are 20 different building blocks, you still have a linear sequence. If you have 4 amino acids attached together, you have 160,000 possibilities. If you have 5, you have 3,200,000 possibilities. Okay? So we're getting an increase in the number of possible molecules generated from a linear combination of amino acids. And this looks like a large number, and it certainly is. I'm not going to argue against that. But it's not really terribly large. It's a tractable problem. And so people have actually developed technologies to sequence proteins. So now mass spec works very efficiently to sequence proteins. And there are still problems being worked out, but this is a tractable problem now and can be accomplished in various laboratories, service laboratories. Likewise, one can synthesize peptides using automated peptide synthesizers. I can order a sequence, they'll make it, and send it over. There are also a wide availability of enzymes to manipulate protein sequences, to cut them, as well as now to paste them, although more advances are really required in this area. Chemical tools, there are a number of chemical tools. These are commercially available in many cases, although, of course, more work is required in this area. So if I mention the term proteomics, I remember 10 years ago when I first heard the term proteomics, I thought it was a ridiculous term. But now, again, this has really penetrated into the popular consciousness of at least biomedical researchers, if not the general public. But I think even now it's reaching down to the general public, and they have an appreciation for the term proteomics and what it suggests. So now if we go on to look at glycans, which is the topic that I'll be focusing on, is you have glycans are composed of principally 10 monosaccharides within mammals. There are many other different types of monosaccharides, of course. I'm not going to go into those. And these can assemble into linear or branch structures, okay, which is different from amino acids assembling into linear structures in nucleic acids themselves. So you get these kinds of complex branch structures like this, shown here. And these are attached onto proteins to form, in this particular case, a glycoprotein. These are known as glycoconjugates. The glycan, the chain of sugar residues, are attached to some other molecule found within the cell. They may be lipids, they may be proteins, or they may be attached to two different things, like a protein and a lipid, for example. Okay, so now these glycan structures, these are not as well defined. They're fairly flexible, and so that makes them more difficult to study using biophysical techniques in certain aspects. Furthermore, if you consider a sequence of 4 monosaccharides that can be assembled in either branched or linear structures, furthermore, they can be attached in many different ways. Amino acids, they only have 2 ends with which they can be attached. Saccharides have many different points at which they can be attached to each other. The consequence of this variation in the way that these saccharides can be attached, as well as the number of different saccharides that exist, makes one face the tremendous possibility of diversity of structure. Okay, so for 4 monosaccharides, one has 7.6 million possibilities, and for 5 monosaccharides attached together, there's 2,633,000,000 different possible structures. Of course, not all of these are found in humans. In fact, there appears to be quite a limited repertoire in humans, but in other organisms, people continue to discover, as well as in humans, continue to discover new types and structures of glycosylation. The primary problem presented by this tremendous structural diversity, which is very sobering, is that it makes the development of analytical tools and methods of synthesizing these structures in an automated way extremely difficult. Now, on top of that, I mentioned that proteins and nucleic acids are directly encoded by the genome. That's not the case for glycans. They're not directly encoded by the genome. They're encoded by the presence of proteins in certain regions and the relative positions of these enzymes inside the cell, as well as the specificity of these enzymes. And so the code is somewhat sloppy, and it hasn't actually been deciphered in many cases about why glycans are built up in a certain way, and what is the code, and how is it well defined. So these are outstanding problems in the area. In some cases they've been solved, and in many cases they have not, however. So when we look at Glycan Technologies, I can't really show you any pictures of commercial products because there just aren't any. People are working. It's not for lack of effort. It's because the problems are extremely difficult. So sequencing, there's really no method for reliable sequencing of glycans. If we look at synthesis of glycans, laboratories have to do this, and it's a laborious process. It's extremely challenging. The chemistry is difficult, and each laboratory has to carry out the chemistry to synthesize the structures that they want to study in a biological context. There are being efforts made to generate automated synthesizer, and these have been met with some good success, and hopefully in the near future we will really see some commercial success in this regard, because that will be a big stimulus for the field of glycoscience and glycobiology. The enzymes, there's a limited availability of commercial enzymes to manipulate glycan structures, and generally it requires a specialist's level of knowledge to use these in an appropriate way. And likewise with chemical tools, there are very limited commercial chemical tools. They're not highly refined, and they require specialist knowledge to use these. So there are some serious problems in the area of glycobiology, and I think now when I use the term glycomics, most of the audience certainly will not have heard of the term glycomics. And there's a good reason for that, because these technologies really don't exist. Ultimately, we would like them to, as researchers in glycoscience and glycobiology, because they will facilitate a tremendous amount of research. So it's my sincere hope and the general hope of the glycoscience community that these technologies do evolve and develop, and that will stimulate further understanding into these areas of human biology and general organismal biology. So with these great challenges, though, come a lot of opportunities. And so this is something that I really want to emphasize, is that the nice thing about being in an area where there are significant challenges associated with studying systems is that there are great opportunities really there, because many systems have not been studied. An understanding of the systems is incomplete, and so a tremendous effort is required to focus on these things. Okay, so continuing on from there, I'm just going to show you some of these diverse glycan structures. Shown here are 4 different glycans in various levels of complexity. So you can have just a single saccharide unit attached to a protein. I'm going to talk about this later on. You can have these complex branch structures attached to proteins. You can have branched structures which are attached to linear structures which are attached to proteins, or you can have linear structures with multiple sugar units attached. Now this is just really a small sampling of the number of different glycans that are out there. There's tremendous diversity in glycan structures. There are many others out there, and it is likely that there are many others to be discovered. I think pretty much every month probably a new glycan structure is discovered in some organism. Okay, now this diversity in glycan structure, why is it important, and what does it really accomplish? Why is this diversity present, and does it have any effect? I think probably one of the most elegant examples of the importance of diversity in glycan structures is illustrated by the variation in blood group antigens. You probably all know of the blood group antigens. You know if you're type A, type B, type AB, or type O. And of course, the discovery of the blood group antigens and the scientific effort dedicated to elucidating the role of the blood group antigens, which involved a concerted effort by chemists, biologists, surgeons, really stimulated and changed the practice of medicine in many ways. Because you can't take blood type A and transfuse someone who has a blood type of B with blood type from blood type A. It just doesn't work. What happens is you have a terrible reaction inside the vascular system where all the blood coagulates, and that results in death. Okay? So you really need to understand what is going on. Now, the structures that define these different blood groups have been defined. And you can see that there are actually quite subtle differences. So this is type A blood group, and this is type B blood group. The only variation between these two is that here you have this acetamido group, and here you have a hydroxyl group. So it's just a small number of atoms that are different, but it has a profound biological consequence associated with this variation. Now type O lacks completely this saccharide residue here, and is just simply a disaccharide. Okay. So going on from there, these blood type antigens are found on the surface of red blood cells and white blood cells and all kinds of tissues. And that really is the basis for this rejection by an immune system of a foreign type of antigen. Okay, I just alluded to the fact that these glycans are found on the surface of cells, and really that is the case. So primarily these glycans are found on the surface of cells. They decorate the surface of cells, and just shown over here is one particular cell, and this gray region that surrounds the cell is actually known as the glycocalyx. And this glycocalyx is composed of saccharide residues that are present on the surface of the cell and reach out into the surrounding environment. Of course, you have this tremendous diversity found in the glycocalyx. All kinds of different glycan structures are present. And what is this glycocalyx doing? Whoa, what happened there? Let's go back. Okay, so these glycans coat the surface of the cells, and we can zoom in and look at the structure of the surface of the cell, a cartoon of it, and these red blobs here are the glycans which decorate proteins and stick off into the solution. Okay? So because they're present on the surface of the cell, they're one of the first things that other cells interact with. And in fact, the presence of these glycans actually enables cells to interact stably and enables the proper development of organisms. So this is a very important process. The presence of these glycans is very important for development. But what's interesting is while it's beneficial for development and essential for development, bacteria and viruses have recognized that these sugars which are present on the surface of the cell are great things to come and stick to. And by sticking to them, they can anchor themselves and then ultimately colonize a host and or invade the cell. So the problem is that viruses also recognize these glycans, and bacteria also recognize these glycans. And so that presents a problem. And this is actually one of the things that leads to this functional glycodiversity, is the constant competition between the organism and these pathogens. You need to change these structures up, prevent these pathogens from ultimately capitalizing on the presence of these and killing off the organism. So there's this constant evolutionary race that goes on. Okay, so these structures, I showed you these chemical structures before with the vertices drawn in stick form. In the glycobiology community, these monosaccharides are generally depicted by simple symbols like these diamonds and circles, etc., which are attached to each other. Now, there are different types of glycans found on the surface of cells. There are N-glycans, O-glcNAcs, such as these, glycolipids, where sugars are attached to lipids, and GPI-anchored proteins, and these are all found on the surface of the cell. Now, why are all these types of different glycans present? I mentioned this before. They're very important in development. They're involved in all kinds of different biological processes, and many of the enzymes that encode proteins involved in transferring these sugars are absolutely essential for life. You can't delete these genes because it results in the death of the organism. The organism never gets born, simply. Okay? So here they play important roles, but biomechanical roles in cartilage. The aggrecan aggregate, for example, is an important component of cartilage. They play important roles in protein quality control and the localization of proteins. So they control localization of proteins as well as the quality of proteins that are being produced within every cell of our body. Likewise, they're also involved in inflammatory processes. So these glycans can signal cells to adhere to certain places where inflammation is occurring. And there are many other biological processes. I'm not going to go into all of these. But suffice it to say that glycans are important in numerous biological processes, and likely many still to be discovered. Now, how are these structures built up? Well, the structures are built up in the following way. So you have a sugar donor here, in this case a UDP sugar donor, and this is used by an enzyme to transfer the sugar part of it onto a target. In this case, it's a protein. So the sugar gets transferred onto the protein, and now you have a sugar attached to the protein, making a glycoprotein. There are over 200 different glycosyl transferases within humans. And some organisms have more. Of course, many other organisms, simpler organisms such as bacteria, have many fewer, of course. Now, this is simply an illustration of how they're attached one after the other in a sequential manner. That's how they're built up. And this structure shown here is the Sil-Lewis X antigen, and that is involved in the inflammatory process, chronic inflammation, etc. So this is a target of considerable interest for the biomedical community. Now, just as these structures are built up, they also have to be broken down. And in humans, there are over 100 different enzymes that break down these glycan structures. They need to act on specific monosaccharides and particular linkages. So shown here is a sugar attached to the protein, the same one I showed before. An enzyme known as a glycoside hydrolase cuts this bond right here to liberate the sugar and liberate the free protein. Likewise, it's a sequential, stepwise manner, the glycan gets degraded. What's interesting is about 2% of the human genome is dedicated to enzymes involved in glycan processing, as well as proteins involved in binding glycans. And I think this really illustrates the important biological roles of glycans, many of which have yet to be uncovered. So shown here, this is a list. It's not intended in any way to confer information other than the fact that there are a lot of entries in this table. The point of there being a lot of entries in this table is that mutations resulting in the deletion of each one of these genes, shown here, these proteins, results in a genetic illness. Okay? So these are genetic illnesses associated with a failure to build up appropriate glycans. Now, there was this nice paper published now in 2002, quite a nice piece of work, where they found actually a genetic abnormality in a person. They were unable to make mannose, and so they were able to treat this person by providing mannose orally to them, and that ameliorated their symptoms very significantly. And this just highlights how an understanding of glycobiology and what is going on at the chemical and biological level really can have a dramatic impact on human health. Likewise, there are deficiencies in enzymes involved in degrading these glycan structures, and there are many of these as well. And shown here is another work published in 1997. This was a proof of concept in which animals, in this case mice, that developed a disease like Tay-Sachs, humans suffer from a disease known as Tay-Sachs, which is a lysosomal storage disorder, arising from the accumulation of glycans to an abnormal level in lysosomes. And this disease has tragic consequences. So they found actually this molecule acted as a compound that could block the pathological accumulation of these gangliosides in tissues. And ultimately that resulted in the development of this therapeutic, Zavesca, which is used to treat lysosomal storage diseases, particularly Gaucher's. Now this is an abnormal cell in which byproducts are accumulating within lysosomes, shown here, these engorged lysosomes inside the cell. And this is a normal cell where the lysosomes are very tiny. And that's the basis for these lysosomal storage disorders. Now, influenza, I think we're almost past influenza season now. Not quite, but almost. Influenza, of course, is an interesting story because its life cycle intimately involves glycosylation. And in fact, the HN code which is used, so H1N1, actually defines the hemagglutinin and the neuraminidase as type 1 antigen and type 2 antigen. The H and the N refer to specific antigens found on the surface of the virus particle. And there are multiple different types. Now, this electron micrograph shows influenza virus particles budding out of a cell where they go off and they're going to infect other cells. And these particles actually bind to the surface of cells using this hemagglutinin, which is a protein that recognizes a particular sugar residue known as sialic acid or neuraminic acid. And the second antigen found on the surface of these virus particles is neuraminidase. And this is an enzyme that actually cuts off the sugar unit, allowing these cells to bud out and spread out to infect other cells. Now, a work published, this is a classic paper now published in 1993. This group headed by Mark von Itzstein in Australia, they were able to develop a compound that acted as an inhibitor of the neuraminidase. And this ultimately led to the development of a compound or therapeutic you may have heard of known as Relenza, which is an anti-flu therapeutic, which is quite effective. There's also Tamiflu, which may be better known because it's orally available, although this is inhaled. Okay, so now I'm going to get on to the projects that we're working on in the laboratory for chemical glycobiology here at SFU. There are 4 projects. I'm not going to talk about all of them. This is one we're studying, this O-GlcNAc modification, and this is what I'm going to focus on and talk about in the next little while. We're also studying carbohydrate processing virulence factors in collaboration with Al Borston at University of Victoria. We're studying O-glucosylation of epidermal growth factor repeats of Notch. This is an interesting project that just started up fairly recently in the lab and is being spearheaded by Garrett Whitworth. And we're also studying AMP-C antibiotic resistance pathway in various gram-negative bacteria that are pathogens. This is work that's being carried out in collaboration with Brian Mark and was spearheaded by Keith Stubbs, who used to be in the laboratory. Okay, so this is a picture of the group. And again, I'd like to acknowledge the tremendous efforts of this group of people. And it's a great, I have a great time working with them, very talented and motivated group of people. Their names you're going to see throughout the talk in the bottom right-hand corner of each slide as I talk about the work. Okay? I'd also like to thank at this time financial support from the Canadian Foundation for Innovation, Alzheimer's Society of Canada, the Scottish Rite Charitable Foundation, the CIHR, NSERC, and the National Institutes of Health Research supported the work that I'm going to be talking about in the next little while. Okay, I mentioned a while ago that glycosylation is found on the surface of cells. And I didn't really tell you the whole story because back in 1984, Gerald Hart discovered intracellular glycosylation. And what they found was that a single saccharide residue could be found on the serine and threonine residues of proteins found inside the cell. So this was really a paradigm-changing discovery, and that was published in the Journal of Biological Chemistry. It's a seminal piece of work now. And it's a very interesting modification because it can act in a signaling capacity in an antagonistic manner with phosphorylation. So this O-GlcNAc modification is illustrated here. There are 2 enzymatic processes involved in the installation and the removal of this sugar residue on proteins. So you have a protein with a serine or threonine hydroxyl group. An enzyme known as OGT or O-GlcNAc transferase acts to install the sugar onto the hydroxyl group. So you have the sugar attached via this beta-glycosidic linkage. A second enzyme known as OGA or O-GlcNAcase, in a more descriptive manner, acts to clip this sugar off, cleaving that glycosidic bond, freeing up the sugar, and liberating N-acetylglucosamine. And this is a dynamic process. And what that means is that the sugar can be installed and removed several times during the lifetime of a protein, which is very much akin to phosphorylation. All right, so there are various ways of studying this O-GlcNAc modification. You can manipulate it genetically. You can also manipulate it with chemical tools, which has certain benefits and advantages. And that is a focus of our lab. We're interested in manipulating O-GlcNAc transferase, shown up here, and we study that enzyme extensively. And if you inhibit that enzyme or block its function, you expect the levels of the sugar-modified proteins to decrease. Likewise, if you block the function of O-GlcNAcase or OGA, you expect the level of these sugar-modified proteins to increase because the sugar can't be removed. But this O-GlcNAc transferase will continue to install the sugar on various sites. Okay, so now the first thing we did when we started research into O-GlcNAc is we started studying the enzymes involved in O-GlcNAc processing. And particularly, we started looking at OGA or O-GlcNAcase. Carried out a number of mechanistic studies to determine how this enzyme actually carries out the chemistry to cut the sugar molecule off of proteins. And that's described in a number of papers shown down here. There are a couple of others. We elucidated the mechanism, And the residues involved in this process, where you take the protein is illustrated by R in this case, the sugar is clipped off, this bond is broken, and what happens is that this group down here actually attacks that center and kicks it out. So the intermediate in this process, in this enzyme catalyzed process, is this bicyclic O-GlcNAc species. And in a second step of chemistry, what happens is this species gets broken down because water attacks here and kicks that group out. And so now you end up with the N-acetylglucosamine. And the protein is liberated earlier on in that cycle. So recently, in collaboration with Professor Gideon Davies at the University of York, and it's a long-standing, very enjoyable collaboration with Gideon Davies and his co-workers there, they determined the structures of these species and found, in fact, that they are as proposed, that you actually have this conformation, which is illustrated by here, which sort of is shaped like a boat. The intermediate, which one can envision this is called a chair. You can look at it as maybe like a beach chair. You sit down on it. In fact, that is the conformation of this intermediate. And this was trapped using various chemical methods that were developed by Keith Stubbs and Matthew McCauley in the lab, as well as Ian Gregg. So a knowledge of what this looks like and this intermediate looks like really allows a definition of what's going on during the process of catalysis, what's happening in the enzyme active site. So we can actually look at that, and this is all of these structures and images are provided by Professor Gideon Davies and his colleagues at the University of York. What's happening during the reaction is actually, it's very simple. This group up here, which is the anomeric center, which is that one, moves from a position above the plane of the ring to a position below the plane of the ring. And so this really simple process defines the chemistry. That's what's going on. It's just this simple motion of the anomeric center, where once it's below the plane of the ring, it's trapped by this acetamido group to form a bicyclic O-GlcNAc species, which is not illustrated in this particular image. Okay, so these biochemical studies revealed the mechanism of this enzyme, and we found actually that it was similar to another human enzyme, another mammalian enzyme, that's found in a different part of the cell. Okay? The O-GlcNAcase is found in the cytosol and in the nucleus, but this other enzyme is found in the lysosome, and it works in a similar manner. If we're going to develop chemical tools, we don't want to inhibit both of these enzymes, so we want to specifically target the O-GlcNAcase. So this presented a problem, but one which ultimately we're able to solve. So using this biochemical knowledge that we had developed in the lab, Garrett Whitworth and Matt McCauley synthesized and tested this compound and found it was a very potent and selective inhibitor of the human O-GlcNAcase. But it wasn't a potent inhibitor of the lysosomal enzyme. And the reason for this is illustrated in this nice image shown here. I'm just going to click on it because it's a movie. And you can see now what we have is a superposition of two different enzymes. We have the lysosomal enzyme shown in gray, and we have the cytosolic enzyme shown in yellow. And you could see this inhibitor, which we call N-butGT, bound into the active site of the yellow enzyme, which is the O-GlcNAcase. It fits very nicely into the active site because there's this pocket there that accommodates this longer chain. However, in the lysosomal enzyme, what you can see is there's this gray residue in the active site, which provides a platform and limits the ability for this inhibitor to bind into it because there's this steric conflict between this point and this residue shown here. So this compound is a potent inhibitor of O-GlcNAcase, 600 nanomolar, and that's a very potent compound. It inhibits the enzyme very effectively and blocks its action. And furthermore, this enzyme is 800-fold selective for O-GlcNAcase over the lysosomal enzyme. Now, here's a structure determined by Gideon Davies and colleagues, Rebecca Dennis, in which we see N-butGT bound to the enzyme, and it fits very nicely into this pocket. And this is a schematic showing some of the interactions where you have this enzyme residue interacting with this group over here. Matt McCauley and Nanya Settenbas carried out some biochemical studies to determine actually the pKa of these groups, and they found that this residue here of the enzyme has a pKa of 5.3, meaning that at physiological pH, it is not going to be protonated. Okay. Likewise, this residue here, this nitrogen of the aminothiazoline group of the inhibitor NBUGT, has a pKa of 3.4, so it is also not protonated at physiological pH. And so we thought, well, maybe we could improve the binding by enhancing an interaction in the enzyme active site. What one could do is make this center more basic. If we make it more basic by converting it from a thiazoline ring to an aminothiazoline, shown here, this changes the pKa to 8. So now at a pH of 7, this group is going to be protonated. And so now you have this negative enzyme residue potentially interacting with a positive group of the inhibitor. Now, I don't really mean to explain in detail the chemical synthesis, but I want to show this slide because it illustrates the power of chemical synthesis. Chemical synthesis is really an amazing technique that can be viewed as molecular Legos, okay, in a very complex way. You have these building blocks. In Legos, you have a limited number of building blocks. When you're doing synthetic chemistry, you have a huge array of different building blocks that you can assemble using different strategies. And the real judicious aspect of it involves the appropriate choice of these building blocks and the conditions in which you're going to use to assemble these. So researchers in the lab developed this very concise route to synthesize this proposed inhibitor, which we termed Thiamet-G. Scott Yosua named it in the lab. And it was carried out very efficiently in these steps by Jenny G, Julia Heinonen, and Ernest McEachern, synthesizing that compound. And we found actually it's a very potent inhibitor of O-GlcNAcase. So shown here is another movie of the compound bound to the active site of O-GlcNAcase. This reveals the global fold of the protein as determined by X-ray diffraction methods. Now we're zooming in on the active site, which is present on one face of the protein domain. You can zoom in and see the inhibitor bound in the active site very snugly, and it interacts with various residues found in the active site, most notably the one that we had suspected it would, this residue shown here. And it forms this very close contact with both of these nitrogen groups. Okay, and the reason for that is shown here. This structure revealed actually not much difference between the binding of NBUGT and this other compound, Thiamet-G. There was a subtle difference in that the inhibitor, the Thiamet-G, was pulled slightly closer. So this is the residue shown here that interacts with this group. And the gray compound is Thiamet-G, the yellow compound is the NBUGT. And it's pulled slightly closer to this enzymic residue. And what that does is it results in much better binding. So now we have a 20 nanomolar inhibitor. And it's now 37,000-fold selective for O-GlcNAcase. Scott Yosua tested this compound in cells, and he found it works really very well in cells. You can get an effect at very low levels of inhibitor. So shown here, this is what's known as a Western blot. And this measures the levels of the sugar-modified proteins, this technique. So you can see here when there's no inhibitor, the levels are fairly low. And when you go up, using increasing levels of inhibitor, you get an increase in the sugar-modified proteins. And it turns out actually that this inhibitor has a half maximal response at 30 nanomolars in this particular cell line, which makes it a very potent inhibitor that's useful for biological studies. Now, at the time that I started at Simon Fraser University, this paper was published in Proceedings of the National Academy of Sciences that suggested that O-GlcNAc may be involved in some way with Alzheimer's disease. So shown here are two brains. This is a healthy brain, shown at left. Well, it's not healthy in that it's no longer in one's head, but it's not disease-affected. So this is a disease-affected brain, Alzheimer's brain, in which you see neurodegeneration resulting in atrophying of the brain. You actually have loss of neurons that results in atrophy of the brain. And so there are two pathological hallmarks associated with Alzheimer's disease. There are these amyloid plaques, and then there are these tau tangles, which are found inside the cell. And this paper proposed that O-GlcNAc may be involved in the tau pathology. So it's known that in the brain, the major nutrient is actually glucose. It's taken up into cells, and it enters into this hexosamine biosynthetic pathway to make the donor sugar UDP-GlcNAc, which you've seen before. I'm not going to dwell on this too much, but this is then used as a substrate by various transferases, including OGT, and OGT transfers the sugar onto tau. Now, tau has O-GlcNAc and it has phosphate. And when the cell is healthy, it was proposed that the balance between these two is maintained in an appropriate manner. However, what can happen is, through environmental effects, diabetes, or the accumulation of A-beta, all of these things impact on the transport of glucose into the cell, and that results in a decrease in the amount of UDP-GlcNAc, limiting glycosylation of tau, and enabling increased phosphorylation of this protein. That was the proposal. It's known that actually hyperphosphorylated tau results in the formation of toxic tau species, leading to neurodegeneration. Supporting this view, the authors of this paper, Chengxing Gong and Fei Lui, found that AD brains have lower O-GlcNAc levels, and tau aggregates have apparently no O-GlcNAc at all. So the normal view of tau hyperphosphorylation is this. Well, you have this is normal tau. It's got a certain amount of phosphate on there. Some pathological processes occur, and that results in hyperphosphorylation of the tau and its aggregation to form these paired helical filaments, which then undergo further aggregation. So a lot of people have focused on making kinase inhibitors, and the objective there is if you block this enzyme that adds the phosphate group, you can block the formation of these hyperphosphorylated tau species. Okay? So that's being pursued extensively. Now, the realization that O-GlcNAc may play a role forces a revision of this hypothesis. First of all, you would then have a more complex scenario. You have hyperphosphorylated tau here, but you have tau with some phosphate and some sugar residues being stable and soluble, and you can have tau with more sugar residues, and presumably this is also going to be soluble and stable and not aggregate. So the idea that we had was, well, what if you actually could use this inhibitor, potentially, to block this enzyme O-GlcNAcase and limit the removal of these sugar residues? Because these sugar residues and phosphate groups can alternate at different residues, you might be able to limit the phosphorylation in that way and not have to affect kinases, which are involved in many important biological processes. And some of the work that Matt Macaulay had been carrying out, looking at the role of O-GlcNAc in insulin resistance, really made some of these studies possible. Because, of course, if the levels of O-GlcNAc result in insulin resistance, this would be a strategy that would be ludicrous to undertake because you would develop insulin resistance. So Matt Macaulay's studies really supported that this may be a feasible route. Unfortunately, don't have time to talk about a lot of Matt's very nice studies. So Scott Yuzwa carried out a number of studies in animals and rats, showed that the inhibitor actually increases O-GlcNAc levels, and also showed that if you treat with the inhibitor, the levels of phosphorylation at certain sites actually decreases significantly. That's shown here in this bar graph. So at certain sites it decreases, and interestingly, at one site it was found to increase slightly. So Xiaoyang Shan and Scott Yuzwa carried out some further studies and looked actually using histochemical techniques to look at the level of glycosylation of proteins inside brain tissue when treated with the inhibitor. And you can see there's a dramatic increase in levels of glycosylation and a dramatic decrease in the levels of tau phosphorylation at this amino acid residue 231, which is a pathologically relevant site, as well as 396 shown over here. Okay, so basically I'm going to leave it there. We're carrying on studies to really try to understand the role of O-GlcNAc in neurodegeneration, to a greater extent. Those are ongoing studies. I think it's really beyond the scope of this talk here, and hopefully we're going to have some exciting results in the near future as Scott Yozua and Xiaoyang Shan carry out those studies. We collaborate with a number of different people around the world. I'm just showing a small number here. Gideon Davies, Yuan He, Carlos Fleitas Martinez. I left out his middle name because it was ending up to be too long. It's shameful of me. And Rebecca Dennis. They carry out the structural glycobiology, studying these O-GlcNAc processing enzymes in complex with these inhibitors and various chemical tools that we develop in the laboratory. Barry Honda and Hugh Brock, Don Sinclair, and Monica Szyzetska. I think that's right. It's hard to pronounce Polish last names, they're tricky. And Tara as well, for collaborating on O-GlcNAc fly genetics. I didn't have an opportunity to talk about that. They have obtained some really beautiful results recently that were published and are really going to shake up the field of O-GlcNAc, I believe. Brian Mark, Misty Balcevich, and Aziza, we collaborate with them on the AMP-C antibiotic resistance pathway. Al Borston on the streptococcal virulence factors that are glycoside processing enzymes. And his colleagues, Charles Krieger at SFU and Chengxing Gong, we look together at some of the O-GlcNAc biology. I didn't discuss any of that as well. Okay, and I'd like to thank you for your attention.
Thank you, David. We do have time for some questions. If anybody has any questions, we would like you to use the microphone as things are being recorded. Are there any questions? No questions? Well, all right, please join me again and as we thank David for a great talk today. Thanks very much, David.